Hey everyone, Mr. Murphy here. I'm going to be taking a look at Chapter 5, which is all about cell membranes and signaling. In this first section, we're going to be focusing on the cell membrane itself. Later sections will talk all about diffusion, osmosis, different types of transport of both small and large molecules. And then we get into all sorts of signaling and how the cell membrane and internal components of the cell handle the delivery of messages, basically from outside of the cell, to cause changes within the cell. The chapter opens with this question of what role does the cell membrane play in the response to caffeine? This is just a good example question of basically how do all of these different things come together in our bodies or in a cell's mechanism for responding to an external chemical stimulus, because that relies upon transport, getting that signal basically into the cell, and then signaling leading to the response ultimately of it. So it's a good kind of broad question to just bring all the different components of the chapter together. Okay, so starting off with the cell membrane, this should be a phrase that we are familiar with and should know and understand the components of, which is that the cell membrane is known as a fluid mosaic, and this is referred to as the fluid mosaic model, which is basically a way of understanding the main characteristics of the membrane in a helpful way to sort of better visualize the mechanisms. So when we say fluid, first off, a strict definition of what a fluid includes both liquids and gases. Fun fact, basically something which is capable of flowing. Now the cell membrane itself and its constituent components is not, strictly speaking, fluid, but in this case it's referring to the changing and moving and capable of the different phospholipids can actually move around within it. The constitution of just like what proteins are present is always changing and can be different from cell to cell or region of a cell to other regions. So it's in constant flux. And then the mosaic component, a mosaic is something that's like a bunch of constituent pieces to form like a large picture. If you look up like mosaic art, for instance, you'll get a sense of how it's many different things coming together to basically create this very unique surface. This tapestry. And here in the textbook, it refers to these phospholipids forming a sort of lake in which the proteins are floating. Again, these are actual chemical molecules being held together, and there's all sorts of intermolecular forces and things like that holding the integrity of the cell membrane together. But this is an effective way of thinking about it as rather than this rigid thing, the cell membrane itself is quite flexible and susceptible to interactions with nearby things or stuff going on in the inside. And the proteins, you really can think about them as sort of bobbing along. And we'll see why later on in the chapter, why it makes sense chemically for those proteins to stay sort of bobbing along in the case of transmembrane proteins. So here you can see what we're talking about, this sort of wave, and nice pictures like this sort of give you a little bit of the sense of that three-dimensional. So this is a big flat plane of phospholipids. You can see different cholesterols and proteins and these little carbohydrate groups sticking off of different proteins and things. You can also see different components of the cytoskeleton here. I mentioned that the cell membrane is not a very rigid thing at all. It is possible for cells to have a very strict and adhered cell shape, which would be facilitated by the cytoskeleton and maybe anchoring with the extracellular matrix, which, remember, is the collagen fibers and things that are outside of the cell. So we've gone through a lot with membranes, and so this section should hopefully be mostly review for us. But just to reiterate, there's hydrophilic and hydrophobic regions. The hydrophilic regions are electrically charged heads. That's the phosphate group. They associate well with water molecules, which are also polar. They also have the interior portion, which is this center bit, and this is where all the fatty acid tails are. So you can always remember that, because the fatty acid tails, we should be thinking fats, and that should help us to remember that that's going to be the hydrophobic or the non-polar region of the cell membrane. And that's why the tails line up together, is because it creates this non-polar, they have these non-polar interactions van der Waals forces and all that this becomes a stable and therefore well-liked orientation for those molecules, if they like things, which they don't. Bilayer, referring to the fact that it is 2 layers. I think we're good with that. And this organization actually helps these membranes. So we're going to sort of bring Chapter 4 and Chapter 5 together when we look more closely at the endomembrane system, but that's the sort of step-by-step process of going from the nucleus, sending instructions out to the ribosomes, to the ER, to the Golgi, to vesicles, to the cell membrane, or elsewhere. And that exchange of membrane material, we'll see how the bilayer sort of facilitates that. That material that was once part of the ER, like phospholipids that were once part of the ER's membrane, may actually end up as part of the Golgi. They may end up as part of the vesicle. They may get to the cell membrane, and then maybe through phagocytosis or something, they get taken back in and they end up sort of recycled again. So if you followed the journey of a single phospholipid, it's very possible that it could end up on all sorts of different organelles over its entire lifespan. And that's again, due to the very specific chemical structure that evolution has led these things to, that it facilitates this very workable system. So in class, when we first introduced membranes, we discussed how the composition of those membranes will depend upon some characteristics of those fatty acid chains. That if it's if it's unsaturated versus saturated, that's going to alter how those phospholipids stack together. It's going to alter how their flexibility, their characteristics at different temperatures, things like that. So the whether it's saturated or unsaturated is going to be one of the most primary, if not the most significant factor in determining the characteristics of those particular phospholipids. Secondary to that would be the length of the fatty acid chain, and then also a factor would be the different kinds of polar groups. So while it's nice for us to say, oh yeah, it's phospholipids, that's what it's made out of, don't be surprised when you continue your career into cellular biology and suddenly you realize, like, curse you, Mr. Murphy, there's not just one kind of phospholipid. There are many different types of lipids, which are constituent parts of a variety of different types of cells and different cellular components. So it definitely does go deeper, but that doesn't mean that this isn't a good sort of avenue for us to understand it at this point, because once you have a firm understanding, like this is sort of the mantra of the course, once you have a pretty good understanding of like the general workings of these things, you can throw all sorts of specific examples or exceptions, and it'll just kind of make sense that as long as you have a good basis for why things are generally the way they are, then you can identify how, oh yeah, this little change, that makes this other thing possible or whatever, and you can understand it in the context of evolution and all that good stuff. So, 2 other factors. Well, we already said lipid composition, but temperature is going to be another thing that's very important for the fluidity of a membrane. And so in terms of like the evolutionary adaptations for different organisms to maintain their own internal their own internal temperature regulation, warm-blooded versus cold-blooded and things like that, that's going to alter their ability to rely upon certain types of membranes to consistently provide certain characteristics, whereas other organisms, and certainly smaller microorganisms, are going to have alternate means of ensuring that the membrane's doing what it should do, even in a potentially wide range of temperatures. Within the membrane, we have all sorts of different types of proteins, and 2 big types are the peripheral membranes, which these are going to be ones that are just sort of on one side or the other. So if I have a bilayer right here, we're going to have peripheral membranes that might be sitting on the top. That's we'll say the exoplasmic, or the outside of the cell side, and then we might have peripheral membranes that are hanging out near the polar head groups of the cytoplasmic side, the inside the cell side. These will lack any sort of hydrophobic groups, because if they have hydrophobic groups, they'll probably be more embedded into the membrane, because the hydrophobic part will play well with the hydrophobic fatty acid tail region of the cell membrane, and so it can be a little bit more thoroughly embedded or even transmembrane all the way across the membrane. integral membrane proteins are partly embedded into the layer, as I'm describing. A transmembrane then would be the third type, which this would be, imagine you have a protein here in front of me, and where my hands are would be like the hydrophilic amino acids, and then somewhere in the middle you'd have sort of a stretch of hydrophobic amino acids. Now this would be a protein that if it was inserted into a membrane would kind of like to just sit there, and it might bob up and down, but it's gonna stay in place because the middle, which has non-polar hydrophobic amino acids, remember the R groups can be any number of different things. So if we have a whole bunch of amino acids in the middle of this protein with R groups that play well with non-polar inside of the membrane, then it's going to hang out there. Meanwhile, the more hydrophilic or polar or charged amino acids are going to be on either end and playing well with the hydrophilic side of the membrane and also the aqueous environment inside or outside of the cell or organelle. So that's going to be a protein that's chemically and physically situated to just sort of hang out and bob up and down inside of the cell membrane, and it's going to want to stay there because of all the chemical interactions. If it starts moving too much one way, then the hydrophobic side starts reaching an aqueous environment, and that's no good, and the hydrophilic side starts reaching the non-polar environment, and that's no good, and sort of pushes it back down. And that's why we say it sort of bobs along. It's like it's floating, it's just chemistry that's causing it to be trapped there, not buoyancy. So some of these membrane proteins can move freely, others are restricted, and they're restricted through other protein or carbohydrate or lipid interactions. And so there's anchoring that can happen in order to keep certain proteins in certain places. There's other mechanisms where basically you create a little pocket of membrane that's surrounded maybe by other types of proteins. So anything that's in the middle of that pocket can bob around, but it can't escape that pocket. There's lots of different mechanisms by which the cell is able to sort of trap and keep certain proteins in certain places. And if you're wondering like, well, why would we want to do that? A good example would be like epithelial cells inside of our intestines, which very specifically have an apical side that is, or an apical side that is pointed towards the inside, the lumen of the intestine, and then they had the basolateral side, the other side, that's pointed towards like your capillaries and stuff. Well, that's going to be two very different sides of that cell, and there's going to be very different proteins. You know, on the apical side, you're going to have the proteins that are facilitating like the uptake of monomers from the inside of the intestine, and on the other side you're going to have proteins associated with delivering ions or other molecules to the bloodstream. So that would just be an example of how there are times when a cell does not want to just have sort of uniform random distribution of proteins and things, and there are mechanisms for that. Do we need to know these specific mechanisms? Not really, but as long as we understand how chemically that would be even a possibility, you're going to be in pretty good shape. So here's a cool example where basically they spliced a human cell and a mouse cell. No, this is not creating some sort of human mouse. It's just individual cells. But looking at these cells and their constituent membrane proteins, the different green ones and red ones here, and then when the hybrid is formed, initially you're going to have sort of this component. Now I don't know the exact mechanism by which they just like karate chop these cells in half and then smashed them together. It was probably something a little bit more nuanced than that. But here you can see initially there's this split 50-50. Realistically, maybe these things were blown apart and then allowed to come back together, but still the human proteins and mouse proteins could be tracked. So maybe it didn't start out split 50-50. But the point is that over time, if these things were tagged, and there's different types of like fluorescent microscopy tagging of cells and cell parts that you can do in order to like actually visualize the movement of these things. Regardless, what they saw was that given time, these proteins actually distributed themselves randomly throughout this cell, which then demonstrates the fact that these proteins are mobile. They are not locked in place. Again, some proteins can be, but in this experiment, it yielded some results that supported the idea of membrane fluidity. So the plasma membrane also has a couple other parts, carbohydrate-based parts, and they can be glycolipids, which is a carbohydrate that's attached to a lipid molecule, or glycoproteins, which are carbohydrates that are bound to a protein molecule. So it sort of depends upon what the function is and all of that, but those also serve important roles in signaling and various other things within the membrane. So just to wrap up, membranes are constantly changing. They're transforming into other types. They fuse, they break down, they exchange from internal cell components to outside cell membrane components. And even though they may look similar, because we always just focus on the phospholipids, just understand that each membrane is going to be a unique mosaic of these different pieces that provides the specific functionality of whatever that cell part or cell is. That's the end of part 1, and next up we'll get into diffusion. Let me know if you have questions.